Where do you think this field is going? And you think the maturity of this is a couple of years away? Are we on that trajectory?
I mean, I think it's going to be a process, right? I don't even know what mature means because I hope we continue to get better and better.
There's too many biological clocks.
Yep.
What's your take on them? Do they work?
Yeah, so here's what I would say about the biological aging clocks. I think the idea that we can measure parameters that can predict your current health status is for sure. One of the things I struggle with is I think the way biological age is used right now is very confusing because what most people really mean is either your risk of dying or your health status.
Welcome to Biopeak Live, brought to you by Longevity India, where we speak and discuss all things health and longevity in the Indian context. I have with me my co-host, Deepak.
Hello. Hello, everyone. I'm Deepak, and I'm the co-host for the Biopeak Live podcast. And today, we have Matt Kaeberlein, a veteran in the aging and longevity community. And Matt, it's a pleasure to host you here. And thank you so much for coming all the way to India and Bangalore, where the air is so clean and nice.
No, I'm loving the year here, guys. I don't know what Brian Johnson's problem is.
So let's come to what Matt's little bit of introduction. Matt is CEO of OptiSpan, co-founder of Aura Biomedicals and he is also an affiliate professor at University of Washington at Seattle. He's probably one of the best known persons in the aging and longevity community. He runs an extensive podcast channel where people have heard a lot of ideas about longevity.
Yeah, it's been one of my staples.
Yeah.
Thank you.
And he's done a lot of interesting research areas, been Rapamycin, mTOR, dietary restriction, signaling. I think we're looking forward to a very exciting session with you here today. Matt, welcome once more. And Prashanth.
Yeah, so my first question from my side is, has it been 3 decades? When did you actually start using the word longevity for what you do?
Well, that's a good question. I'm not sure when I started using the word longevity. I started studying aging as a graduate student. So I went to graduate school thinking I was going to do X-ray crystallography or protein structure or something like that. And in my first year, I heard a talk by Lenny Guarenti, who was a professor there, where he talked about how his lab, just in the last few years, had started studying aging and using genetics and molecular biology. And I had never thought about, I was in my, late 20s probably. So I'd never thought about aging really that much anyways, but certainly not that you could use it as or that you could study it as a biological problem. And I just got fascinated by that idea and joined Lenny's lab and started working on aging in yeast at that time. I never looked back, really. I think what evolved in my thinking was, the recognition that while aging is a really interesting biological problem, the impact, if we can modulate aging for human health and companion animal health, I'm very interested in having an impact on the health and lifespan of dogs and cats and other companion animals as well. But the impact from targeting aging is so much greater than the impact from targeting individual diseases that that really has, motivated me to continue in my career for, almost 30 years now. I was 1997 when I started graduate school. So a lot has changed in the field in that time.
You're probably the first part of the first cohort, right?
Lenny's,
that lab seemed to be like kind of the fountainhead.
Yeah, the way I think of it is, aging research goes back certainly to, the early 1900s. If you think about the work of people like Clive McKay and others on caloric restriction, the way I think of it is the time I came into the field was a really exciting period because it was really, I feel, the transition from sort of phenomenological observational work, molecular biology, genetics, biochemistry, and like you said, mechanism focused. And so the people like Cynthia Kenyon and Gary Rufkin and Lenny and Linda Partridge, all around that same time, started applying these tools in very mechanistic ways. And I think that it was exciting for lots of reasons, but it shifted the view of the broader scientific community to start to view the biology of aging as a credible scientific subdiscipline. Even still, it was sort of looked at as a backwater. There were lots of questions about the quality of work, but because people started to use these mechanistic approaches, I think we gained credibility in the scientific community. Yeah. It was also fun because that was a time when people started to do what I call large unbiased genetic screens for longevity. So, using C. elegans in yeast primarily, we could start to look at many, many different genes and just ask the question, which genes affect aging? And so it felt like every week somebody was discovering a new gene that impacted aging. And so that led to many of the initial pathways being filled out, the insulin signaling pathway and mTOR and sirtuins. And we can talk about sirtuins if you want. There's some stuff there. But it was a really exciting time and fun time in the field. And then, I've been around long enough that I've seen, the kind of next paradigm shift, which I would characterize as the hallmarks of aging becoming sort of the dominant view of the field. And I think that's also been good and bad, in many ways. And, now I think we'll see, are we going to have another shift with AI coming into the picture and, really changing the way we think about the complexity of aging biology?
Matt, the question which I want to know from you, you said you bumped into longevity by listening to probably Lenny.
Yeah.
And then it became a passion. Then your trajectory is interesting. You seem to have moved away from academia and moved into setting up companies and trying to explore the corporate culture. Why you moved from academia?
Aura, we spun out of my lab when I was still at the University of Washington. So that's Aura Biomedical. And I'm obviously still involved with Aura, but I'm not day-to-day involved with Aura. So I kept my academic position. So I think there were many reasons, I think, as there always are for especially for somebody who makes a pretty significant career change after, at that point, 25 years, being in academia. I think some of the things that contributed were, as many people in academia feel, frustrations around constantly having to fight for grant funding, feeling like your best ideas don't get funded, yet you get funded for things that are sort of pedestrian and incremental. I think everybody in academia probably has felt this at one time or another. So that was a constant frustration source. And I was very fortunate. I was well funded. I never went through a period when I was in academia where I really was concerned that I would run out of funding for my lab. But it felt like I spent so much of my time doing that it wasn't being spent on things that were important. And then I think also just the sort of administrative creep that happens as you get more senior. So I went into science because I love the process of scientific discovery. I'm a very curious person. I love the feeling of discovering something nobody else has ever known before. I sort of lost that. And then, and I don't think I've ever told this story before, so I'm not going to use any names, but I think the moment I knew I needed to look for something outside of the realm of academia, I was at a conference, and, like is often the case in a relatively small field, which longevity was at that time and to some extent still is, but growing rapidly, is the same people giving the same talk, meeting after meeting. Yeah. And so I've heard these talks, a lot. And so I'm kind of just half daydreaming, looking around the room, and I look at like 3 or 4 of my colleagues who are 20 years senior to me, and I literally asked myself this question. I'm like, do I want to be them in 20 years? And I don't mean that in a way like anything bad about them, but the fact that they were still struggling to write grants and still fighting the same battles. And the answer was immediately clear to me, no, I do not want to be doing this for 20 more years. So I knew that I needed to be open to other possibilities. So I didn't immediately decide, I've got to get out now, but I just became open to other ideas and looked around. And the other thing I think that really weighed on me that helped crystallize that decision was in my career, I feel like there have been a few situations where an idea that I had, I knew was the right thing to do. I knew it needed to be done. And one of those was the dog aging project. And that was back in 2014 or so. And I had to fight so hard for 5 years to get enough funding. And it wasn't just me. Daniel Promislo, Kate Creevy, we were all doing this together. But we had to fight so hard for an idea that is so freaking obvious it should be done. I just didn't want to go through that again. And I felt the same thing was kind of happening with what we ended up doing at Aura with the WormBot. And again, happy to talk about any of this in detail, but that frustration that in academia, big ideas that will actually move the field are really, really hard to get. So all of those things just made me think, okay, I need to do something else. And so I did.
No, I enjoy what you said, but it's giving me some scary thoughts also. I'm coming from a academic background. Yes, I know where it goes.
Yeah, look, that was the right decision for me. I'm not one of these people who comes out bad. There's a lot of good stuff that happens in academia.
I think the best word which Matt captures is academic frustration can actually drive you nuts if you're not really getting the right clicks. I mean, for us, I would actually say Longevity India was a crazy idea for us. Prashanth happened. It still stuck around.
Hey, that's fantastic. I'm really impressed.
So, Matt, I think people can't talk about you without talking about Matt.
Sure.
It's good and bad. So
yeah.
But
I've gotten to the point where I have to say, like, I have no financial interest in Rapamycin. Yes.
Maybe you should have had that.
I know, right? If I did, I would be able to fund my own research one day.
Yes. So maybe you should have thought about the spin out early.
Yeah.
But anyway, I think, but on the other hand, that of all the mechanisms, that influence on the mTOR pathway, still the central aging influencer, and nothing much has changed in the field.
Yeah.
So sometimes do you feel that there has been now a phase where we are going through where there has to be one more big fundamental unlock? Maybe AI will make that happen, but like what the MS did when it came to protein folding, or there's some such initiative, concerted effort, a lab that now kind of unlocks using AI, the next big thing.
I think that's likely the case. What we know for sure is that when we simply look at our ability to modulate the biological.
Maybe that makes sense for others. Just talk about mTOR and just a little bit. Yeah.
Sure, I'm happy to do that, but I'm gonna start more broadly.
Yes,
and because I think mTOR is clearly a key player in the network that we know about that affects aging. But more broadly speaking, if you just look at our ability to impact aging, and probably the best way to do that is to look at how much can we increase lifespan, right? In any animal, but in particular in a mammal. So we're probably talking mice in the laboratory. To my knowledge, the largest effect on lifespan, taking out sort of changing genes and having animals go through development in a different genetic state, is an experiment that was done 50 years ago by Weindruck and Wolfer, a caloric restriction experiment.
Yeah.
And rapamycin in an mTOR inhibition are kind of second on the list, but it's only about half the size of the effect of that caloric restriction experiment. So why aren't we continuing to get bigger and bigger and bigger effects? I think that tells us that we're missing something, right? We should have done better than caloric restriction in the last 50 years. Now and again, I say that not because I want to diminish what has been learned, because we've learned a lot about the biology of aging, but we haven't fundamentally gotten past that barrier. So that makes me think there is a barrier that we don't really understand why we can't do better than that. Probably it's at least in part because all of the targets that we're going after right now, mTOR, insulin signaling, AMP kinase, are in that same part of the network.
Yeah.
And we need to figure out what don't we know. That's fundamentally the way I would characterize it. I think we know what we know, and we don't know what we don't know, and we don't know whether what we don't know is 10% of aging or 95% of aging. I suspect it's more like 95%
of aging. Yeah.
So, and I think I mentioned a couple of minutes ago that I think the hallmarks of aging have been both good and bad for the field. I think one of the ways they've been harmful is that as soon as the hallmarks of aging were formalized, it became very difficult to study anything that didn't fit into that
their
paradigm.
team of things, yeah.
And so people stopped looking. I mentioned also earlier that when I came into the field, people were doing lots of unbiased genetic screens. That all pretty much stopped about the time the hallmarks became formalized. Nobody's looking outside of the hallmarks. I think we need to. I think there are a few approaches that should be taken. I think one is comparative biology of aging, looking in animal models that haven't been studied yet. I think one is high throughput longevity intervention screening, and that's where tools like the WormBot and Aura can come in. And I think one are these, very large data discovery science projects where the new AI tools can help us to understand that complexity and maybe, like you said, unlock something fundamental that we don't understand right now. And the nice thing is, I think we're really starting to see momentum towards that third approach, right? People taking large population-based multi-omic sorts of approaches, and then hoping, and I expect this will happen, that the AI tools will continue to develop and help deconstruct that data in really meaningful ways. And I should say there's a lot of optimism around things like epigenetic reprogramming, to be determined. Maybe that will be the unlock we're looking for. Again, I'm not particularly confident that it will be, but maybe that will be. But I think there's something fundamental we don't understand yet about the biology of aging. And I'll just finish up by saying I was just before I came here at a conference in Singapore. It was what do they call it? Gerophysics conference.
Yeah.
Physics and geroscience. And I think that was really interesting because there are some people who are taking very mathematical, quantitative approaches, very simple in terms of the math that was being used, and already starting to tease out potentially interesting insights into, for lack of a better way of saying it, different dimensions to aging. And I think most of what we've been studying is only one dimension, and there's at least 2 or 3 or 4 dimensions that are out there that we haven't yet been able to understand.
So, Matt, one of the more recent efforts, and this is from the same Nemesis team, is to build a virtual cell.
Mm-hmm. Yeah.
And they believe that, to really model a complex, dynamic logistical system itself is a prerequisite before you can apply AI, because you just can't apply AI on just a lot of random data.
Absolutely. Yeah.
So anyway, so I think that's so these unlocks are something else.
And I think in the aging field with AI, there have been people who've been trying to use AI tools, for years in the field. I think the challenge has been what you were alluding to, which is that our current sort of understanding of the system is relatively crude, and it's a garbage in, garbage out sort of situation. And when it comes to the interventions, this is one of the reasons why I've argued pretty strongly that we need to explore the intervention space for longevity much more deeply, because you can't use the AI tools when you only really have 3 or 4 things you're confident robustly extend lifespan. Yeah. You can't really use AI and predict what else is going to extend lifespan.
That is very true. I think it's pretty fascinating what you said.
There's a common
denominator there.
And it's a
So
topic of
you've got OptiSpan, and OptiSpan, as far as you understand, want to use AI data and combination thereof. You said something very fascinating. The viewpoint of hallmarks of aging actually narrowed our view so dramatically that we refused for a very long time to think beyond this conventional norm. And I think this use of multi-omic, multi-parametric, and large data-driven approaches are definitely going to open up the next dimension of these facets of aging. Where do you think the new dimensions will come from? Because if you have ticked the boxes of saying inflammation on, stem cell exhaustion, this wrong, is there something where do you think something beyond this picture will come from?
So I think there's a couple of things that will happen for sure. I think one is we'll find more things that we could characterize as hallmarks. So the list could get longer.
Okay.
That's going to happen. I expect where the AI tools will be very powerful in the short term from these multi-omic data sets is helping us understand why it is and how it is that the hallmarks are interconnected. In other words, what is the structure of that underlying network, right?
That network thing is
Yeah. So I think that will happen. And I mean, again, I'm not saying anything super profound, but I think those are fairly obvious extensions of where we are now. What I wonder is whether or not we will start to get clues to, sort of what in my own mind I think of as the dark matter of aging. Like, again, I am just more and more starting to believe there's something fundamental that we have missed, that's out there. And I don't know. I think that this sort of physics approach is going to help us become more confident that that's there. I don't know that it's going to help us figure out what it is. And so maybe the marriage of those two things, the AI large data approaches and these sort of modeling, more basic mathematical modeling, will help us kind of figure out what that is. And then we can start to try to intervene in this second dimension, for lack of a better way of saying it, of aging. So I think that's an unknown, but I think there's a chance that could happen. I would also say this is why I don't think we should put all of our eggs in one basket, so to speak, and just hope that if we measure enough stuff and use AI, we'll figure it out, right? So that's one approach to try to do that. Another approach is, again, to take a step back and just let the biology tell us what's going on, right? And this is where I think the large-scale intervention screening should happen. So again, this may be based on the fact that, like I said, when I came into the field, that was kind of what revolutionized our understanding of aging. But I think what most people don't appreciate is we stopped early. So those large-scale genetic screens, even when you just look at the genetic landscape, is a tiny fraction of the genetic landscape. These are mostly, single knockdown or knockout mutations across the genome. So you can't query essential genes. There's nothing on increasing function. They were all knocking down function of genes. So nothing on increasing. And if you think of it like a drug, those are all single dose experiments, right? We knock the gene down by a certain percent. So there's this huge landscape, even genetically, we haven't explored. And then when you look in the small molecule space, we've done almost nothing.
Yes.
So another approach would be to just say, if we could measure 1 million, 5 million, 10 million interventions and find out which ones affect lifespan, we're going to find new things. I am 100% confident we're going to find things that are better than caloric restriction, and we can actually start to look at combinations. And I think that's an area where the field has really lagged. We have very little data on what happens if you combine 2, 3, 4 interventions. Sometimes you get interesting, surprising effects. And again, maybe this is just my bias being a sort of curious science guy. Like, I like to be surprised. I like it when you do an experiment, you're like, that is not at all what I expected, right?
Yeah.
So we will find things that are unexpected. And those unexpected discoveries, in my experience, are the ones that fundamentally change your understanding.
So to keep your head open for those.
Yeah.
So one quick question. This fantastic narrative you've given about that we have not screened enough. We have not tested enough. We have not really explored the dimension, which is So one of the big question coming from an academic perspective and also from industry, what's the best model system for aging to screen?
Well, so yeah, I think that I'm glad you said that, because I think what's the best model system for aging really depends on what your use case is, right? So, and for screening, it depends on the scale you're interested in. And obviously resources. If resources were unconstrained, in principle, you could do 1 million intervention screen in mice. Now, even there, you'd have to really ask yourself, is sacrificing 50 million mice worth it, right? I mean, that's a different question, but it would also take you years, decades, really. So the way I thought about this was, when we were thinking, okay, first of all, one question is like, what number of interventions would you need to test to really move the needle? 10,000, 100,000, 1 million. And one way to answer that is to ask, how many interventions do we know about? And that's hard to answer precisely, but you can look in the largest database that's out there. It's called Drug Age, and there are about 1,000, right? So it's a pretty small number. So, 100,000 would get you pretty far. So that's kind of where my thought process started. This was, 10 years ago now. And then you have to say, okay, if I want to measure 100,000 interventions, I'm going to do this using small molecules rather than genetics, just because, it's easier and small molecules are interesting. Where could we actually do that? Where is it feasible to test 100,000 interventions? And so you're limited, if you're not going to do it in mice, to fruit flies, C. elegans, or yeast, really. Those are the 3 model organisms that have been used in the field, with some frequency. Killifish starting to be used because small molecule delivery in killifish, really tricky. I didn't actually know this. I was talking to someone at the meeting in Singapore. They actually have to go in with tweezers and stick it in the fish's mouth. So that's probably not doable. Very tedious. So thinking about this, I really thought, okay, we have to automate it. And so in what system could we automate? And so that kind of takes fruit flies out of the system. This is getting a bit technical, but you have to change the vials and all that.
Yes.
So it'd be hard to automate. So C. elegans was the place we landed. And we were pretty confident we could build a robotic system that would mostly automate the lifespan experiments to take the human equation out as much as possible. We did that, recognizing that C. elegans are not perfect, right? There's lots of reasons why stuff that works in C. elegans might not work in mice and people. As it turns out, I think it's pretty good going that direction. I think most things that work well in C. elegans, and by well, I mean large effect size. That's the great thing about screening 1 million or 100,000 interventions. You don't have to study the stuff that has tiny effects. You can study the stuff that has big effects.
Yeah.
So I think it works pretty well going that direction. I think what you lose in C. elegans, you miss a lot. And this, again, is the vagaries of C. elegans biology, but they just don't respond well to some drugs. And so you're going to have a fairly high false negative rate. You just have to live with it. So that's how we settled on C. elegans. We built the WormBot. It's a robotic system coupled to AI that mostly automates the lifespan experiments.
The million molecule challenge, how was it related to it?
Yeah. So the million molecule challenge was my response to being, one of my responses to being frustrated by not being able to get this kind of a project funded in academia. So again, to me, this was a no-brainer. If you could test 1 million interventions for a few $1 million, we waste millions of dollars. I shouldn't say that. We spend millions of dollars on projects that are tiny incremental projects. But NIH reviewers have been trained to be very conservative and only fund incremental research. We, the scientists, have trained ourselves to do this. It's a feature of highly hyper-competitive funding, not wanting to fund a grant that's going to fail. So regardless, you could never get a project like that funded through NIH. And the first response you get is it's a fishing expedition, meaning you don't know what you're looking for. You're letting the biology, you're doing discovery science. And my response is, well, you're never going to catch any fish if you don't go fishing. But NIH reviewers don't buy that. So I was frustrated by that. And so we tried to we spun Aura out of the lab as a for-profit company. And at the same time, trying to go out and raise investor funding for Aura, wanted to create this community opportunity. It's go to the community and say, if you think this project is a good idea, you can pick the intervention, cost you $100. We'll test it. We'll put it in an open access database for anybody in the community to get access to. And believe me, that was my goal from the very beginning. I wanted to create a large open access database with 100,000, 500,000, 1 million interventions, because I think that data set seeds a whole bunch of discovery science throughout the rest of the field, including really smart people who know how to use AI much more effectively than I do to query that data, and then in a more targeted way, start to tease out the patterns and tell us which interventions are likely to work. Then you can start to use AI to tell you, well, maybe if you combine these 4 things, it's going to give you a 4-fold increase over one of them alone. So that was the idea behind the Million Molecule Challenge, was to create this opportunity where anybody in the world could sponsor interventions to contribute to this open access project.
Is it still live?
It's still live. Yeah, it's been, I don't want to, I didn't have any preconceived notions as to how much the project would raise. So, but it's been a little bit disappointing to me that it hasn't raised as much as, I had hoped it would. Again, I sort of feel like I did what I could. Like, you present it to the community. If people are going to buy in, they're going to buy in. If they're not going to buy in, they're not going to buy in.
So one interesting thing, Matt just mentioned about middle molecule, and you've been saying that we need to find something which is better than caloric restriction. And caloric restriction is a mixed set of physiological changes which is happening in your body.
Yeah.
So do you think one molecule can meet that kind of complex requirement? And especially coming from a country like India, where we talk about a mixed formulation always tends to work better. We always talk about systemic drugs.
Yeah.
So like classic example is our curcumin. The pure molecule just doesn't work, but the herbal extract of turmeric, which is the source of curcumin, does seem to have anti-inflammatory properties. So are we looking at the right way for a million molecules, or we need to look into more mixed formulations?
Well, I would first of all say we want to do both. Like, I don't think, even though it's called the million molecule challenge, the intention was never to make that as a million
individual molecules. Yeah, great.
In fact, that's the beauty of this system, is you're not constrained by the number of things you can measure. So you can test combinations. So yes, I don't know whether a single molecule can be as effective as caloric restriction. My intuition is yes. I think we just haven't found the right molecule and or the right dose. Certainly you're not going to have as many biological effects as caloric restriction with a single molecule. What we don't know is, if you think of all of the different things caloric restriction does in a living animal, how many and which ones are important for the lifespan extension.
Yes.
If it's 10,000, then you're probably not going to get that with a single molecule. If it's a few, you might. And I think rapamycin is a pretty good case example. We can come back to mTOR and rapamycin because we never really did a deep dive on that. But rapamycin is a drug that inhibits mTOR. mTOR is a sort of key nutrient sensor in every eukaryotic cell that we know of in the world. And one of the things caloric restriction does is it potently turns down mTOR. And rapamycin is a drug that does the same thing. Now, caloric restriction does about 10,000 other things. We're probably more like 100,000. So rapamycin is only hitting a subset of the biochemical changes that caloric restriction is hitting, but you get about half the lifespan effect, the best lifespan effect we can get from caloric restriction. So is there a dose of rapamycin or a different way of inhibiting mTOR that would get you closer to caloric restriction? Probably. Would you get all the way there? I don't know. But I think you could probably get pretty close.
But it's always interesting that whenever you look at more small molecules used for aging, you keep hitting the insulin signaling pathway. Because we are still trying to mimic caloric restriction.
Yes.
But why don't we hit some other pathway? Is there a windows between narrow, or this is the pathway?
It's a good question. I don't know the answer. I'm pretty sure it's not the only pathway. And I think there's a selection bias, clearly, that has gone into many of the studies over the last 15 years, right? Again, this is getting back to the hallmarks, not only the hallmarks. I mean, scientists tend, part of the reductionist mechanistic approach is you tend to dive deeper and deeper and deeper and study the processes that you think are important, for good reason. Like, I'm not saying that's the wrong approach, but I think you also want to take this more unbiased approach from time to time to help you figure out what you don't know, what you don't understand. So that could also explain why we keep hitting the insulin signaling pathway. I will say, if you look at the data from the unbiased genetic screens, there are lots and lots of genes that came out of those screens that don't seem to map to the insulin signaling pathway. It's just that nobody has really studied them and figured out how they're working. I think the closest was a project that we did in yeast that took 15 years from start to finish. Really, this is Brian Kennedy and I who started this. And it was the sort of end of that project where we tested the lifespan of like 5,000 gene deletions in yeast was finished up by a professor at New Mexico now named Mark McCormick. And he did a really good job of kind of putting these things into buckets and categories. And it turns out, I don't remember, there were like 7 categories that we could put most of the genes into. Not all of them were related to nutrient sensing. So I think there's a lot out there that just hasn't been followed up because it's not what everybody's following it. Yeah.
And looking at rapamycin trial here in India, we've been kind of thinking about it. We just wanted to get your perspective on learnings. Have you done anything in tropical countries before? Is there any aspect of that picking on mind? And second is just coming out of the Pearl trial and learnings from there.
Yeah.
I think we probably should take cognizance of a few things and set it up right.
Yeah, I would definitely agree with that. So the tropical country piece is interesting. I can't think of any trials or data sets from off-label use of rapamycin that have been derived from tropical countries. The only one that might be close is I believe that
Singapore is doing something.
That's what I was going to say. I believe that I don't know how many people have been treated or if that started. That there is something going on in Singapore. Obviously, Brian would know about that. So the things I would think about. So again, one of the questions is, one thing I would think about is if and whether mTOR inhibitors have been used in organ transplant patients in India. That's a literature that you could look at and just ask.
It has been extensively.
So is there any difference in side effect profiles in that population?
Again, not something which you look, but we know the cases of infection does go up in those people. That because of the dose differences.
But the question would be, does it go up more in people in India versus people in other parts of the world? That data is probably out there.
Yes, it should be something which you can worth looking into.
So that would be one place to just because I think the question is, would there be an increased risk of things like infections?
Yes, that's the.
And so if you don't see it in organ transplant patients, then I think you probably don't have to worry about it.
Not I think so, because the dose is quite high.
Right, exactly. And we know that those people are at higher risk for infections. We don't really know in people using it off label. And I'm using that term. I guess I should be a little more precise. Most people using rapamycin off label, and all that means is prescribed by a physician for something other than the label.
Right?
Yeah. So most people using rapamycin off label use it differently than it's used in organ transplant patients. Most organ transplant patients are taking a few milligrams daily. Most people using rapamycin off-label are taking somewhere between 3 and 6 milligrams once a week.
Much lower dose, yeah.
And there are some interesting differences in the pharmacokinetics that may lead to lower side effects. I think most people would say it's pretty sure bet now that off-label use, both because of the lower total dose and the once-weekly dosing, has a lower side effect profile. So the question is, in those people, what do the side effects look like? And the honest answer is nobody's done the randomized clinical trial, so we don't know for sure. But we did a study where we collected survey data from 300 some people who'd used rapamycin off label and 100 and some people who'd never used rapamycin to try to get a feel for, what does the risk look like? And so one of the ways we assessed side effects was we said, in the last 3 months, have you experienced X? And X was a long list of potential side effects, including bacterial infection, viral infection, whole bunch of stuff, pain. And what we found was that there were, I think, 7 things that were statistically different between the groups. The only one that was higher in the rapamycin users was mouth sores. That's a known side effect in organ transplant patients. In organ transplant patients, those are pretty severe ulcers. In the people using it off label, it was more like a typical canker sore. But that was, I don't remember how much, 7 or 8-fold higher in the rapamycin users. The other 6 things were all lower in the rapamycin users. So who knows what it means? Infections, bacterial infections were trending a little bit higher in the rapamycin users, but it did not reach statistical significance. So I don't know what the answer is. My guess is if there is an effect on bacterial infections with off-label use of rapamycin, I believe there probably is. It's pretty small. So
that's interesting.
So that's the information that I've got.
And what's the scale that you would look at if we had to do some analysis?
You mean how big would the clinical trial need to be?
How much does it have to be powered?
Well, that really depends on what your endpoint is, right? So, what I can tell you is there are a few clinical trials happening now for different indications. So one is a clinical trial for periodontal disease. So we had shown in mice that rapamycin can reverse periodontal disease. Jonathan On, who was the graduate student in my lab who did that work, is now doing a clinical trial in people. So for that, I think they're looking at, 40 or 60 people. So it's not a huge trial.
The one in New Zealand.
What's that?
The one in New Zealand.
Yeah, Brad Stanfield's got his clinical trial.
Point there?
That is muscle function as measured by, I think, a sit-stand test and maybe grip strength.
Grip strength.
Yeah. And that's also relatively small. I will also say, though, this is pretty typically the case in these kinds of trials. They're often not really solidly powered for detecting efficacy.
True.
They're often branded as a safety trial. And again, this is one of my frustrations with the academic system in general. People do this because reviewers will fund these small quote unquote safety trials.
Yes.
And the investigators will do this because then they think that if it doesn't cause any problems, they'll be able to get another grant in another 2 years to do, a little bit larger trials. So it's an incentive structure that leads to underpowered clinical trials
And
that
incremental
takes
gains.
many, many, many years to ever get to an answer. So I dodged your question because I don't know the answer, but it does depend on what the endpoint is. My guess is if you really wanted to have a rock solid clinical trial, you need a few hundred people, just ballparking it.
So staying to the topic of clinical trial, let's say you're trying something rapamycin, you're trying a rapamycin metformin combination or rapamycin metformin urolithin combo, which could be very easy and tempting thing to try.
Yeah.
What were the markers you would look for? What would be your bet on, say, 5-10 markers I will look for to see if there's some kind of even incremental gain in quality of life if given choice?
Right. So the things I think about, well, first of all, I would say I don't think I'd include metformin, but that's just my personal bias. If I was going to do something to try to hit glucose regulatory control, I'd probably use an SGLT2.
Better, yes.
So that's a different discussion.
Yes.
And I know different people have different opinions on metformin. So I think the things I would think about, obviously, you'd want to look at the kind of standard blood-based biomarkers. I'd want to do a deeper dive on inflammation because we have good reason to believe that particularly rapamycin is gonna knock down chronic sterile inflammation. One of the things we've been doing at OptiSpan, and it's a little bit of a discovery process, is looking at some of these these blood tests that are specifically for autoimmune disease and asking whether within even the reference range, we might be able to detect early indications of chronic inflammation, obviously CRP and things like that as well. So I'd look there. I'd look at body composition for sure. So you mentioned the Pearl trial. That trial ended up having a lower dose than they wanted to deliver because they used encapsulated compounded rapamycin. But even then, there was a hint of improvements in body composition in women. This is an anecdotal thing that I've seen over and over, just from people telling me that rapamycin may facilitate weight loss and preservation of lean mass. Actually, thinking of combinations, I'm super interested in a trial of a GLP-1 agonist and rapamycin at the same time. Yeah. So I'd look at body composition as one of the things that I would monitor. Infection, obviously, because we talked about it. One of the things there I would be interested in is looking at both bacterial infection and viral infection. So my intuition is, like I said, maybe a slight uptick in risk of bacterial infection and probably a larger decrease, not in risk of viral infection, but risk of downstream severity of the viral outcome. And one of the reasons I say that is another thing that came out of that study I mentioned where we looked at rapamycin users was the use period this was right after COVID that we started the survey. So many of the people who took the survey were using rapamycin during the pandemic. And so one of the questions we asked was, did you have a diagnosed COVID-19 infection? And if so, we had them rate the severity according to specific criteria, and then also asked them if they had prolonged symptoms, something that might be long COVID. there was no difference in the risk of having had a COVID-19 infection between the users and the non-users. But when we looked at severity, the users were significantly less likely to have a moderate or severe outcome, and also less likely to have these symptoms that might be long COVID. So this fits with a growing body of literature that rapamycin can boost antiviral gene expression, and probably also the many of the prolonged negative consequences of viral infections, not just COVID-19, but other viruses as well, are driven by chronic inflammation. And that's exactly what rapamycin is really good at knocking down. So that's another thing I would think about looking at. So those would be the things. And, if you could do it, vaccine response would be really interesting too, because Joan Manick had these early studies with a derivative of rapamycin showing boosted flu vaccines on elderly people. But the reality is, we could sit down and in an hour probably come up with 15 or 20 endpoints that would be interesting to think about looking at. And then you really have to kind of focus in. And
which ones do you?
Yeah. Another one, this is something I recently learned. There's a couple of papers that have come out. I'd be interested in doing brain MRI. So the studies that have come out, they're small. They're not
neuroinflammation suppression.
Yeah. So there's a couple things that are probably going on. And these studies were both in ApoE3, E4 carriers, right? So heterozygotes, they're at higher risk of dementia. They often show early declines in brain volume, particularly in the hippocampus and the caudate nucleus. And in one of the studies, they did MRIs before and after, and actually saw within, I think it was 4 or 6 weeks, increases in brain volume in both of those regions, as well as increases in cerebrovascular blood flow. And the other study replicated, it wasn't replicated because they weren't trying to replicate, but they also saw increases in cerebrovascular blood flow. So it would be interesting to think about whether rapamycin could have an effect there. And now I'm going to speculate wildly just because I can't help myself. So I also had a chance to talk to Greg Fahey recently, and I don't know if you guys know who Greg is, but he developed this TRIM protocol, the thymic rejuvenation protocol.
Growth combination of
growth hormone, metformin, and DHEA. And I am intrigued by the idea of what effect does rapamycin have on the thymus. My intuition is while you're taking it, it's probably not going to do much, but when you come off, you might see something similar to what Drake sees. His sort of. I have absolutely no data to back that up, but I would be interested. If you're going to do MRI, you might as well measure the thymus and
exactly. That's true.
So one last question, and I think we can probably jump to the
quick fires.
Okay.
So Optispan, in some sense, has the vision to be kind of a new age medical concierge or a new age 4.0.
Yeah.
Where do you think this field is going? And you think the maturity of this is a couple of years away? Are we on that trajectory?
I mean, I think it's going to be a process, right? I don't know. I don't even know what mature means because I hope we continue to get better and better. But what I would say is, I think right now, there are real opportunities given the tools we have available when applied in a rigorous and medically sound way to have major impacts on the health of most people. Again, I have some data to back it up, but again, it's a little bit of speculation. But I really think in the United States, most people can get 15 years of quality life back by using a mix of early diagnosis, preventative healthcare, lifestyle modification. I don't think you really even need to go to rapamycin and things like that. Maybe GLP-1s for people where it's the right thing. I think most people can get 15 years back that way. And then the opportunity. So then the question is, can we kind of deploy that in an effective way at a large scale? That's part of what Optispan is about, is what is the best way to use the toolkit that we have now and to hopefully standardize a little bit across the space, what works, what doesn't work, where's the gray area. So I think there are real opportunities now. And then I think going forward, the question is, how do we determine within the gray area, and I put rapamycin in the gray area, even though I've used rapamycin, I'm pretty bullish. The real answer is we don't know with 100% certainty, right? Who's going to benefit from it, who's not, what the benefits are going to be. So start to figure out how do we get sufficient data within the gray area. And I'm not suggesting we're going to be able to say with 100% certainty, rapamycin, yes, urolithin A, no. I'm just picking on urolithin A. I actually am sort of bullish on urolithin A, but really put them into solid buckets. But it's all probability, right? But if we can actually get good data so that the docs in this space are empowered instead of just guessing. And honestly, most of them are just guessing, and most of them are guessing from a position of ignorance. They hear some influencer on the internet talk about X and they start prescribing it to people, right? I think we need more rigorous approaches, and we've got to collect data from lots of sources, and we don't really have a way to do that right now. So I think that'll be kind of the next step.
I'll kick it off with a relative feature on one of our quick fire. 3 supplements that you would really stand by, and 3 that you think are kind of a bit on the overhyped side. Okay. No vitamin D, no omega.
Oh, well, that just took 2 of my 3. I mean, the reality is I think the ones we can stand by 100% are the things that we know are strongly associated, if not mechanistically causal for disease, right? So, and we can measure, right? That's the other thing. If we can't measure it, it's really hard. So, vitamin D and omega-3, I put in the solid bucket because we can measure it. We're really confident there.
The next level, which is
I mean, I don't have a lot in that level, honestly. So personally, what I would use, would be things like magnesium. I know that many people would put that in the same bucket. The issue there is we can't really measure it. So creatine I'd put in there. I think the data is pretty solid, and personally, I feel like I've benefited from using creatine.
So urolithin A and sparvadine.
Yeah. So So the preclinical literature is good. I believe that they have pretty significant impact. The human data is missing mostly. There's a little bit on urolithin A, a little bit on epidemiology and spermidine. I think the question there is not so much, I believe that they can have positive impacts in people. It's more around dosing and which people. So I don't see a huge downside other than, what it costs you, which for some people is significant, right? But but I also am not confident that in the way that these supplements are being developed and sold, that they've got sufficient bioavailability, and we know
if there's enough of a difference that make up.
Yeah, and I mean, I would put a lot of stuff in that category. I would put alpha-ketoglutarate. Again, data's pretty good, solid. Even the NAD precursors, I believe there are many people who have deficiencies in NAD that could benefit from the NAD precursors. When I say many, I don't know if it's 10% of the population or 60% of the population. I'm pretty sure it's not 100% of people. I think part of the challenge with the NAD precursors is they have been so overhyped that, if I have sort of this visceral pushback against that. And so I tend to come off as a little bit more negative about them than
What about the 3 bad ones?
Well, I mean, the one that I have studied that always, sticks up number 1 in my mind is resveratrol. It's not that it's bad. It's just that it was presented as a, game-changing longevity drug, and that has been completely debunked. Like it is the most debunked longevity drug. Yeah, I've said that before. Yes. So again, doesn't mean it can't have some health benefits for some people, but there's just no evidence that it targets the biology of aging or has positive impact on longevity. There's not a lot that I feel strongly like that visceral negative reaction about. I just think there's a lot of things that there's no real evidence to support, and that's like a super long list.
That's true.
I'd put resveratrol in that list too. I just talk about it because it was, it got such a big push attention. Yeah. And I think it contributed to a lot of people thinking that drinking red wine is good for you. Whether red wine moderately is good for you or not, I think is a different question, but it certainly isn't because of resveratrol.
That's true. So it's a standard thing is that resveratrol is going to keep you young because you die young.
Yeah, that's right. That's right. Yeah.
So Last quick fire question.
Okay.
There's too many biological clocks.
Oh, yeah.
What's your take on them? Do they work?
Yeah. So here's what I would say about the biological aging clocks. I think the idea that we can measure parameters, I'm just going to say it that way because they could be molecular, but they don't have to be, that can predict your current health status is for sure. One of the things I struggle with is I think the way biological age is used right now is very confusing because what most people really mean is either your risk of dying or your health status. Yes. So these things, that's what they actually were trained on. They're correlated to either your risk of dying, your risk of getting a certain disease, or your overall health status. All of those things are correlated to biological age, but they aren't identical to biological age. So none of the clocks are measuring biological age, in my view. I'm not even sure what biological age is. I said really good concept. I'm not sure what it is. It's
published in papers.
Yeah. Yes. It's in the titles of many papers. I'm actually going to say this in my talk tomorrow. It's probably been talked about in this meeting 18 different ways, like 18 different things people called biological age. So what I would say is I think there are tools that we can use to predict somebody's risk of dying or somebody's current health status or future health status. And there are a variety of flavors. The one that gets talked about the most are epigenetic age clocks.
Yes.
And I have two things to say about that. One is, as research tools, they are extremely powerful. And I don't want to diminish the potential of these epigenetic patterns to be used to tell us fundamental things about aging, to predict interventions, to predict personal trajectories. I think that there are real opportunities there. And I'm excited to see where that goes. But they have been rushed to market by companies who are selling these tests to consumers, and there's really no quality control there, and they just don't work. The noise is bigger than the signal. And so that has created, I think, a lot of, uncertainties and I think a growing distrust. While at the same time, you're seeing more and more medical providers offer, sometimes recommend these tests to their clients, and they don't know how the tests work. There's nothing actionable from these things, and so it's a challenging time, I think, in the industry side. And I point to the epigenetic test because that's the place where I actually have data. I suspect this is true for all of the different consumer-facing biological age clocks that are out there. Yeah.
Fantastic speed. We covered a lot of things. Yes.
Hope to hosting you in Bangalore again, and I hope you like the air. Yep.
Yeah. I can't wait. I've had a great time and looking forward to coming.
Thanks for being there on this episode of Biopeak Live. Look forward to seeing you all on the other side. Okay.